Welcome back to The Deep Dive. Today we're tackling what is still, I think, one of the most frustrating clinical mysteries of the COVID-19 pandemic.
Oh, absolutely. The sheer variability.
Exactly. The radical variability in patient outcomes. You, the physicians and specialists listening, you're on the front lines managing this whole spectrum.
Right. For people who were totally asymptomatic, didn't even know they had
At
it.
the critical cases in the ICU with multi-organ failure, ARDS, and now, of course, the long tail.
The persistent chronic issues, long COVID, and maybe more worryingly, these completely de novo autoimmune conditions that seem to just pop up.
The inconsistency is just baffling. But it's not random, it's mechanistic.
And that's the key point. That enormous spectrum, I mean, from a sniffle to a complete immune collapse that told us the driver wasn't just the virus's virulence, it had to be the host.
It's rooted in our unique genetic and immunological architecture.
Yes. We have to stop just describing the symptoms and really start digging into the molecular failure points that determine who gets sick and who doesn't.
So our mission today is very targeted. We are going to trace that molecular architecture of vulnerability. We're going to connect what seem like disparate dots.
The genetics, the molecular mimicry, the inflammatory response.
And we're going to show how they all converge on a single central regulatory system. We're going to make the case that the tyrosine kinase or TK signaling network is that central failure point.
The very thing that dictates both the acute severity and that chronic autoimmune risk you're now managing in your clinics. This is really about building an operational map for more personalized immunology.
So the core question we're trying to answer today is this: why? Why is the TK pathway, and specifically we're talking about the JAK-STAT axis and some key receptor TKS, why is it the engine that drives this pathogenic T-cell differentiation and even the neurological damage we see in susceptible people?
Right. We're moving past just talking about the cytokines themselves to the signaling hubs, the factories that produce and amplify them.
Okay, so let's start at the beginning. The story of a severe infection is often just boiled down to the cytokine storm. But the intensity of that storm, it's really predetermined by what happens in the first few hours.
Yes. You have to start at the genetic gates. The initial ease of infection is all down to host genetics, beginning with the front door the virus uses to get in.
And that front door is the ACE2 receptor.
Right, the angiotensin-converting enzyme 2 receptor. That's well established. But the really crucial insight, especially for this audience, is in the subtle single missense variants in the ACE2 gene.
So tiny genetic changes.
Tiny changes that fundamentally alter the efficiency, the binding affinity of the spike protein. I mean, we are literally talking about molecular physics determining your risk.
And what I find so fascinating here in how they figured this out, they weren't just guessing, they used molecular dynamics simulations. They actually measured how these structural changes impact the binding energy.
Yeah, they quantify the electrostatic forces, the van der Waals forces, right at that spike ACE2 interface. They could literally calculate the energetic cost or benefit for the virus to dock.
And that quantification is so critical because it lets you identify the variants that dramatically lower the infectious dose.
Exactly. You can find the genetic changes that give the virus a quantifiable head start. So let's focus on those high-risk variants that were found, particularly the ones that are more common in European and South Asian populations.
So which specific changes gave the virus the biggest thermodynamic advantage?
The single strongest one they found in the computational modeling was a mutant called AC2G211R.
G211R.
Yeah. This single substitution created the largest calculated increase in binding energy. It was something like 7.6 kilocomol, more favorable than the wild type.
Wow, 7.6 kilocomol is a huge advantage at the molecular level. How does that actually work? What does it look like?
It's a classic electrostatic trick. The substitution swaps out a neutral glycine for a large positively charged arginine at that position, 211. And that dramatically stabilizes the whole spike ACE2 complex through these really favorable charge interactions. It's like it locks the virus's key into the cell's lock with much, much greater efficacy.
So for anyone carrying that G211R variant, it probably means a successful infection requires way less viral inoculum. The immune system is immediately on the back foot.
That's the thinking, a much higher early viral load.
But here's a question. If a variant gives the virus such a massive advantage, why doesn't every single carrier of G211R get critically ill? The penetrance still isn't 100%.
And that is the critical point. It's a layered vulnerability.
Yeah.
The ACE2 gate, dictates the speed and the ease of the initial infection.
But not the quality of the immune response that comes next.
Exactly. That's handled by a different set of genes, HLA. And we saw other high-risk ACE2 changes too, like ACE2D206G. In that case, you lose a negative charge, and it seems to recalibrate the local electrostatic field, making the initial binding just that much more favorable.
Again, just amplifying that initial viral foothold.
Right.
And it's important to remember the contrast, too. The sources also found variants that did the opposite, that actively reduced the binding, like AC2K26R.
Yeah, that one decreased the binding favorability by about 2.1 picomole.
So it's a reminder that susceptibility is a dynamic, measurable spectrum. It's not just on or off. For you, the clinician, the bottom line is that these ACE2 polymorphisms really dictate the start of the race.
Whether the infection starts as a trickle or a flood.
Exactly. So once the virus gets past that entry gate, the next crucial step, what determines progression to severe disease and autoimmunity, is the quality and speed of the adaptive immune response.
And that brings us directly to the highly polymorphic and, well, often failing battlefield of the HLA gen complex.
HLA molecules, what we used to call MHC proteins, they're the presentation platforms. They're essential for kicking off the adaptive response.
Right. We know Class I activates our CD8 plus cytotoxic T cells, and Class II gets the CD4 plus T helper cells going.
And the huge polymorphism in these genes, that's what dictates the quality of the T-cell epitope menu that you're presenting to the immune system.
And that menu is everything. The research points to these specific genetic blind spots, HLA alleles, that are unfortunately pretty common in some groups, that are just structurally unfit to grab and present the most critical SARS-CoV-2 peptides.
So the defense mechanism that should be fast and targeted is just subpar from the get-go.
Fundamentally flawed.
So let's talk about the most striking finding here, this dual risk allele that seems to connect the failure in the acute infection to the risk of chronic autoimmunity.
That would be HLA DRB1 08.01. This allele kept popping up. It's been associated with an almost doubled risk of getting COVID-19 in the first place and increased mortality, especially in really vulnerable groups like transplant recipients.
And the mechanistic studies, they provided the proof, right?
They did. They showed this specific allele was, for the most part, unable to bind any of the tested SARS-CoV-2 peptides with high affinity. It just couldn't show the T cells what they were supposed to be fighting.
That failure of presentation is the absolute key point of vulnerability. So why? Mechanically, why does this DRB1 08.01 molecule fail to bind these viral fragments?
It's all about the architecture of its peptide binding groove. DRB1 08.01 has this unique feature, a deep hydrophobic P4 pocket. It's formed by specific amino acids, glycine at P13, leucine at P74.
And that structure determines what kind of peptides it prefers to bind.
And tragically, its preference just does not align with the critical viral epitopes. So if your T cells don't get the right molecular mugshot, the adaptive response just stalls, and the virus gets an insurmountable lead.
And what makes this so predictive, so concerning, is its known link to chronic autoimmune conditions already.
Exactly. We already know DRB1 08.01 confers susceptibility to severe autoimmune diseases like lupus and MS. So you have the direct link from a specific genetic presentation failure to both acute severity and chronic autoimmune risk.
A risk that's probably amplified by the molecular mimicry we're going to get to later.
No doubt.
We also saw some other associations, right, in Italian cohorts for HLA CO1 and B44.
Yes, those were hypothesized to be more permissive rather than outright deficient.
What does that mean, permissive?
It means they might present less effective epitopes, or they have structural restrictions that lead to a kind of fragmented or inadequate T-cell response. It's just not enough to clear the high viral load that was established by the ACE2 variants.
And that's worrisome because, like DRB1A01, these permissive alleles are also often linked to pre-existing inflammatory diseases.
Right. It suggests that in these carriers, the baseline immunological profile is already kind of tuned towards dysregulation. Even before the virus comes along, it pushes it over the edge.
It's such a profound contrast when you look at the protective alleles. What's the success story here?
The poster child for success is HLA B15.03. This allele showed the complete opposite effect. It seems to have one of the highest binding affinities for a whole range of SARS-CoV-2 peptides.
So it's an excellent presenter.
An excellent presenter. It facilitates a rapid, robust T-cell activation and, as a result, gives strong protection against severe COVID-19. This genetic split just underscores that severity isn't random at all. It's the direct result of these subtle changes in the host's ability to communicate an immunological threat.
Okay, so we've set up the opening act. High infectivity because of ACE2 variants, then a failure of the T cell response because of HLA genetics. So once the immune system loses control, the infection escalates, and the resulting immunopathology puts the tyrosine kinase network center stage.
Right. This is the control panel for inflammation and for cellular fate.
The TKs become this central regulatory hub, and they're driving both that crazy inflammatory signaling in the lungs and, as we'll see, the very precise neurological damage through structural mimicry.
We're moving from a genetic predisposition to the molecular executioner.
So let's start with the cytoplasmic TKs. We know severe disease is synonymous with that hyperinflammation triad. Lots of mediators are involved, IL-1, TNF-A, but the signal amplification, the feedback loops.
They're most strongly correlated with extremely high levels of IL-6, MCP1, and PTX3. Those are the ones that really correlated with lethal outcomes.
And IL-6 is really the centerpiece of that cytokine storm.
Unquestionably. And its signaling is fundamentally, completely tied to the cytoplasmic TKs, specifically the Janus kinases, or JKs.
Okay, so walk us through that. IL-6 binds its receptor on the cell surface. And what happens next?
It triggers a very rapid cascade. First, the activation and phosphorylation of JAKs, which are at their core tyrosine kinases. Then these activated JKs go on to phosphorylate the STAT family of proteins, especially STAT3.
And that STAT3 activation is the key event that really propagates the crisis, because it's a transcription factor.
Right. This high IL-6 signaling drives STAT3 into the nucleus, and it kicks off this extensive genetic transcription program that leads directly to the Th17 trait imbalance.
Which is a hallmark of so many chronic autoimmune conditions.
Yes, and this is where we really need to slow down, because the way these T cells get pushed towards that pathogenic Th17 lineage driven by IL-6 and TGFO, it involves a really critical molecular trick. It's not just hyperactivation, it's a permanent cellular change.
You're talking about the non-canonical pathway. How does IL-6 and STAT3 lock that T cell into a pathogenic Th17 fate for good?
It uses an epigenetic lock. So instead of just turning on the standard target genes, the source material shows that STAT3 physically binds to intron 7 of a gene called RFX1.
It binds to an intron, a non-coding region. That's the sophisticated part. What happens when it binds there?
It triggers these simultaneous cell-sustaining epigenetic changes. Specifically, you get a decrease in histone acetylation and a big increase in DNA methylation right at that gene locus.
And the net result of that is?
A functional shutdown of the RFX1 gene. It's silenced.
Okay, so what does RFX1 normally do?
RFX1 is a crucial molecular brake. It's a brake on the entire Th17 program. It helps suppress the genes that a T cell needs to fully differentiate into a Th17 cell.
So by silencing the silencer?
By taking the foot off the brake, the TK-driven STAT3 cascade locks the T cell into a pro-inflammatory pathogenic fate. This is why that cytokine storm is so hard to stop once it really gets going. The T cells are, for all intents and purposes, irreversibly committed.
And that epigenetic lock, it creates this profound self-amplifying system. Once you have these Th17 cells, they're pumping out IL-17.
Which in turn drives massive production of IL-8.
And IL-8 is a potent chemoattractant. It calls in floods of neutrophils to the lungs.
And you get that vicious self-amplifying loop that culminates in ARDS, multi-organ damage, and death.
And we can link this inflammatory susceptibility right back to common host genetics.
We can. The IL-6 174G allele, which is prevalent in over half of European populations, something like 55 to 65%.
Yeah, very common.
Very common. It predisposes carriers to producing significantly higher levels of IL-6 when their cells are stimulated.
Sure.
So you're connecting a common genetic trait directly to the severity of the cytokine storm via this JAK-STAT TK signaling pathway.
And the clinical utility of breaking this cascade is really what validates the whole mechanism.
Absolutely. The rapid adoption and, the proven success of JK inhibitors like buristatinib,
which prevents STAT phosphorylation,
and IL-6 antagonists like tocilizumab, which block the initial signal, that confirms that this TK pathway isn't just involved. It's the central targetable hub in acute hyperinflammation.
Okay, so if the cytoplasmic TKs are governing the storm, the receptor TKs and their counterparts, the phosphatases, they seem to be the focus of the structural damage, especially in the nervous system.
Right, which is highly sensitive to any signaling dysregulation. And here, molecular mimicry is directly targeting the machinery of the TK network itself.
This is a really critical pivot. We're moving from functional inflammation to actual structural compromise.
Yeah, the immune system is being molecularly tricked into attacking the very architecture that regulates cell growth and survival.
So let's look at the first key target.
Yeah.
The foundational receptor TK, ERBB2.
Right, ERBB2. It's part of the HER2 family. It's a critical growth factor receptor family. It's absolutely essential for maintaining neurological tissue.
And the molecular mimicry found here is exceptionally precise, isn't it?
It is. The spike S2 subunit shares a 5-amino acid sequence, a pentapeptide, PLQP, with the ERBB2 receptor, PK.
PLQP. So if the immune system makes antibodies or T cells against the virus's PLQP,
Those same cells will cross-react and attack the host's own ERBB2.
and the implication for the nervous system there is profound. You're attacking this receptor that's essential for survival signals in nerve tissue. Could this be a plausible molecular mechanism for some of the neurological complications we see?
I think absolutely. But it gets even more alarming when you look at the attack on the system's counter-regulator, the break of the signaling network.
And that is PTPRZ1, the receptor tyrosine phosphatase.
Exactly. PTPRZ1 is a phosphatase. Its job is dephosphorylation. It's the direct antagonist to TK signaling. It turns the signals off. It regulates things like synaptic plasticity in the CNS.
So targeting PTPRZ1 essentially means you're creating a state of chronic TK signal overdrive. The on switch is stuck.
And the mimicry here is, it's not just precise, it's widespread. PTPRZ1 share 5 distinct short peptide sequences with the spike S1 and RBD subunits.
5.
Five different opportunities for the virus to trick the immune system into generating antibodies against this vital regulatory break.
So an immune attack against the break implies a systemic immune-mediated dysregulation of the entire TK pathway within the central nervous system.
Leading to chronic hyperactivation.
Clinically, what does that look like for a patient? The TK system stuck in on mode.
It looks exactly like the chronic neurological symptoms we classify as long COVID. Profound brain fog, cognitive impairment, chronic pain, peripheral neuropathy. When you attack PTPRZ1, you compromise the signals that are critical for myelin and synapse maintenance. You lose the ability to precisely regulate neuronal communication.
So this really shifts the mechanism of neurological damage. It's not just viral invasion, it's a highly specific immune-mediated attack on the central signaling control panel.
That's it.
And we're not just relying on computational predictions here, are we? Is there empirical proof of this happening in patients?
There is. Researchers validated a specific spike nonapeptide, VLKG-KKLHY.
Okay.
It shows almost 90% similarity to a human protein called KF14. And KF14 is a critical motor protein. It's essential for axonal transport, literally moving materials down the nerve fiber.
And they found evidence of an immune response to it.
They did. They experimentally confirmed that this specific viral epitope was recognized by CD8 plus T cells that were recovered from convalescent patients. This is direct proof that molecular mimicry is driving cellular pathology against essential neuronal machinery. It's impacting the physical integrity of the nervous system.
This molecular deception, then, where the virus shares these sequences with our own proteins, it provides a concrete mechanism for the huge range of autoimmune conditions we're seeing.
Right. It's a plausible multi-system mechanism. We're seeing type 2, type 3, type 4 autoimmunity, all potentially arising from these cross-reactions.
But before we get to the de novo autoantibodies, we have to talk about that catastrophic pre-existing risk factor that just cripples the innate response right from the start.
The neutralizing IgG autoantibodies against type I interferons.
Right. IFN alpha and omega. This isn't mimicry. This is a pre-existing autoimmune failure that almost guarantees severe disease.
This was a landmark finding. It really helped explain who progresses so rapidly to critical illness. These neutralizing autoAvs were found in at least 10.2% of patients with life-threatening COVID-19 pneumonia.
And the timing is everything.
The timing is paramount. They were often present right at the onset of critical disease, and importantly, they were virtually absent in mild or asymptomatic cases.
That correlation is such a powerful predictor. If you have them, it means your first line of antiviral defense, your innate type I interferon response, is immediately neutralized by your own immune system.
Precisely. You're prevented from switching on that crucial early antiviral state. That leads to uncontrolled viral replication, high viral loads, and that systemic crippling of the innate response is what sets the stage for the massive inflammatory backlash.
The TK-driven cytokine storm.
Exactly.
And this also gives us a clear molecular explanation for one of the most stubborn epidemiological puzzles of the whole pandemic.
The sex and age bias and mortality.
Absolutely. 94% of the patients who had these anti-IFN autoantibodies were male, and the prevalence just went up significantly with age.
It provides a direct molecular basis for that severe skew in who dies. For you, the clinician, this is highly, highly actionable.
How so?
Well, screening for these anti-IFN autoapps when a patient is admitted should be critical. Knowing they exist completely changes your treatment calculus.
Because giving exogenous type I interferons to that 10.2% of the population would be
Clinically futile. The treatment is instantly neutralized. So instead, you need mechanism-specific alternatives, like the TK inhibitors, to target the downstream cascade that's going haywire.
Okay, so let's look now at the massive potential for multi-system autoimmunity that comes from the viral mimicry itself. The computational screen you mentioned found 1,224 T-cell epitopes.
Mapping onto 285 different human proteins.
That's a staggering number. It completely explains the broad multi-system potential for T-cell-mediated autoimmunity.
The immune system sees the viral peptides, and because of these structural similarities, it generates responses that then go on to attack host proteins all over the body. Let's start with the endocrine system. That's a really common site for post-infectious autoimmunity.
Okay, so the data showed that anti-spike antibodies had significant cross-reaction with thyroid peroxidase, or TPO.
And TPO is the classic autoantigen biomarker for Hashimoto's thyroiditis. This finding provides a direct molecular pathway for the rise in post-COVID or post-vaccine autoimmune thyroid disease that clinicians are seeing.
So Hashimoto's, subacute thyroiditis, even Graves' disease.
Right. Clinicians need to have a high index of suspicion for new onset thyroid dysfunction in the months after infection or vaccination.
Okay, moving on to metabolic and neural risk. The anti-spike antibodies also cross-reacted with glutamic acid decarboxylase 65, GAD65.
Yeah, GAD65 is a well-known autoantigen. It's linked to type 1 diabetes and, neurologically, to stiff person syndrome.
So the immune response to the virus could inadvertently create antibodies that target this enzyme, which is crucial for both insulin regulation and inhibitory neurotransmitter function in the brain.
The link there is deeply concerning for future metabolic health.
We hear so much about the profound, debilitating fatigue in long COVID. The research seems to offer a molecular explanation for that, rooted in the mitochondria.
It does. Anti-spike antibodies reacted strongly with mitochondrial M2 antigen. M2 is central to cellular energy metabolism. This gives us a direct link between molecular mimicry and mitochondrial dysfunction.
Which maps perfectly onto the clinical presentation of symptoms like chronic brain fog and that profound fatigue.
The cells simply can't generate energy efficiently because their power plants are under immune attack.
And shifting back to the structural damage in the nervous system, which specific proteins related to demyelination were targeted?
We saw cross-reactivity with myelin basic protein,
Yeah.
MBP, and neurofilament protein, NFP.
Both are essential structural components.
Essential. MBP is a primary part of the myelin sheath, and NFP forms the structural scaffolding of the axons themselves. Attacking these provides a molecular basis for demyelinating conditions.
Like Guillain-Barré syndrome, GBS, or OMAD.
Yes, conditions that reflect immune-mediated damage to the axons in the myelin.
And it's crucial to point out that the systemic risk isn't just limited to T cells or the vascular system. We have to think about barrier integrity.
Absolutely. Cross-reactivity was also found with tight junction proteins, specifically occludin and zonulin.
These are the molecular mortar holding together our critical barriers.
The gut, the lung alveolar wall, the blood-brain barrier. Molecular mimicry targeting these proteins suggests the immune response might compromise these protective barriers, making it easier for inflammatory mediators to get into places like the CNS.
And finally, we always focus on the spike protein, but the mimicry extends beyond that.
It does. The virus's non-structural protein, NSP3, specifically its macrodomain, mimics a human protein called PRP14.
And what does PRA14 do?
PRP14 is a critical regulator of macrophage activation. So exposure to NSP3, which you get in a severe infection or from some whole virus vaccines, could lead to an immune attack on PRP14.
Which would disrupt macrophage control.
And potentially sustain a damaging chronic inflammatory state, especially in the lungs. It's an important point. Mimicry doesn't just target the host, it can target the host's ability to turn inflammation off.
So all of these detailed molecular insights, the TK dysregulation, the epigenetic locking, the structural mimicry, they aren't just abstract concepts. They have to translate into clinical management.
They do. And they help explain some of the otherwise baffling sequelae we're seeing, especially the post-vaccine phenomena.
Okay, let's talk about that clinical challenge. The de novo seronegative rheumatological symptoms reported after vaccination. Things like PMR-like muscle pain, that intractable fatigue, bilateral ankle arthritis.
And with a really significant female predominance, something like 70 to 75%.
And the clinical picture is often atypical, right? It confounds diagnosis because the standard inflammatory markers, ESR, CRP, are frequently normal.
Normal or just minimally elevated. Yet the patients are profoundly symptomatic. It's very frustrating for both the patient and the clinician.
So what was the crucial immunological signature they found in these symptomatic patients?
It was the hyperproduction of anti-spike antibodies. I mean, levels that were often exceeding the assay limits over 2080 BAU/ml. And critically, they persisted.
For a long time.
For a very long time. Up to 8 to 25 months post-vaccine. That long tail of very high antibody titers seems to be the key.
And the leading hypothesis is that this is linked to antigen persistence.
That's right. The persistence of the spike S1 antigen fragment, possibly being retained within antigen-presenting cells like monocytes long after vaccination.
And this acts as a low-level continuous stimulus.
A continuous toll-like receptor, or TLR-driven stimulus.
So it's not a massive acute immune attack, but more like a chronic low-grade irritation of the immune system because the antigen hasn't been properly cleared.
Exactly. And that persistent, low-grade TLR stimulation leads to sustained, chronic production of IL-6 and TNF-alpha. And here's the direct TK link.
Perpetually feeds that JAK-STAT TK hyperinflammatory pathway.
Yes. The system is essentially stuck in a state of low-intensity, high gear. It's enough to cause chronic symptoms, but not enough to trigger those classic acute phase markers like a high CRP.
This mechanism also offers a potential molecular explanation for that strong sex bias we see.
It does. The research showed that the spike protein has a high binding affinity for ER-alpha, the estrogen receptor alpha.
So it competes with estrogen.
Potentially. By competitively binding to that receptor, the spike protein may effectively displace estrogen and neutralize its normally protective anti-inflammatory effect.
And that disruption would be most pronounced in women, explaining the 70 to 75% female predominance.
That's the theory. It would exacerbate that TLR-driven, TK-fueled inflammatory state.
So from a clinical standpoint, how does this guide your differential diagnosis, especially when the inflammatory markers are normal?
It really shifts your focus from just measuring acute phase reactants to thinking about a functional immunological assessment. When you see persistent rheumatological symptoms, but a normal ESR and CRP, the differential has to include this persistent TK-mediated signaling.
And the clinical pearl, the thing that validates this mechanism, is how these patients respond to therapy.
Exactly.
So what response are we looking for?
These patients often respond remarkably well to low-dose corticosteroids. We're talking 8 milligrams of methylprednisolone, tapered quickly to 4 milligrams.
And that response acts almost like a diagnostic confirmation.
It does. It validates that there is a real underlying steroid-responsive inflammatory process being driven by this persistent, low-grade, TK-mediated signaling. Even when the bulk markers of inflammation are normal, it proves the pathology is in the signaling cascade.
So the rapid clinical success of targeting the TK pathway in acute COVID really did validate that core molecular hypothesis.
Absolutely. The adoption of JAK inhibitors like baricitinib and IL-6 antagonists like tocilizumab to manage acute cytokine release syndrome showed us that the JAK cascade is the primary driver of the acute pathology.
And that success really shifts the paradigm toward precision therapeutics. If TKs are the central hub, what other specific modulation strategies does the data support?
The focus has to be on mechanism-specific intervention. The centrality of that STAT3 signaling hub is underscored by research into alternative ways to modulate it.
Okay, let's clarify the mechanism proposed for a compound like ivermectin, purely as a way to probe the STAT3 disruption. How does that fit into the TK pathway?
Well, the hypothesis is that it works by suppressing the degradation of a protein called PAC1.
PAC1.
P21 activated kinase 1. Now, PAC1 is a key scaffolding protein, and it's known to bind directly to both JK1 and ST3. This physical interaction is essential for the signal to propagate efficiently during hyperinflammation.
So by preserving PAC1, how do you interrupt the STAT3 signal?
By suppressing its degradation, you compromise the physical interaction of that whole PAC1-JK1 STAT3 complex. You're basically preventing the scaffolding from forming properly.
So you disrupt the signal transduction needed for that high-volume STAT3 signaling.
Right. It highlights the importance of disrupting that precise kinase interaction, not just trying to reduce the amount of cytokine ligand floating around.
That level of specificity is what we need. And returning to a critical prophylactic strategy, screening for those anti-IFN autoantibodies seems non-negotiable, given the 10% prevalence in severe cases.
It should be, yes. The future strategy have to demand early identification of this pre-existing condition. As we said, giving exogenous interferons to those patients is futile.
But because their pathology quickly becomes a massive JAK-STAT-driven hyperinflammation.
TK inhibitors offer a mechanism-specific alternative for managing that acute phase. You're targeting the downstream cascade and completely bypassing the disabled innate response.
So this integrates diagnostics and therapeutics into a single genotype-informed approach.
Yes, it's about moving beyond a generalized sepsis protocol to an individualized molecular management plan. We're treating the host's specific failure point.
So to synthesize this entire deep dive, we've established that COVID-19 severity and chronic risk, they're not dictated by a single failure. It's a highly layered, personalized process rooted in our own genetics.
It starts at the genetic gates, those ACE2 variants and poor HLA presentation from alleles like DRB108.01.
And that's followed by what you called personalized molecular deception, the molecular mimicry targeting key TK regulators like PTPRZ1.
All of which is amplified by the master regulatory pathways of tyrosine kinase signaling the JAK-STAT axis, which can be epigenetically locked into a pathogenic fate.
I think the key takeaway for everyone listening is that the focus has to shift. We have to move from just describing the clinical syndrome of autoimmunity to identifying its precise molecular address.
Which is overwhelmingly rooted in the TK network. We have identified genetic vulnerabilities that predispose people to both immediate acute failure and this persistent chronic immunological dysregulation.
Which brings us to our final provocative thought for you, the clinician on the front lines, tasked with future preparedness. Given this clear identification of high-risk HLA alleles like DRB1 over 8.01, the IL-6G allele, and the documented mechanism of TK-driven pathology, how rapidly should the medical community move?
How quickly should we be moving toward integrating high-resolution HLA typing and TK pathway markers into personalized risk assessment for future viral threats?
For those genetically susceptible individuals you identify, should this molecular data inform the need for prophylactic TK pathway modulation, maybe guiding the urgency of starting a JK inhibitor at the first sign of exposure?
Or, and this is maybe the most critical question, should this molecular knowledge fundamentally guide the composition of future vaccines? Should we be actively excluding and avoiding these known molecular mimicry epitopes like that PLQP sequence shared with the ERBB2? This is the critical next step, moving toward truly personalized immunological medicine, transforming risk prediction into actual preventative intervention.
A profound and absolutely essential question as we move from generalized protocols to genotype-informed patient management. Thank you for joining us for this deep dive into the molecular architecture of host vulnerability. We'll see you next time.